Good evening, Mr. Chairman, distinguished guests, and fellow researchers. My name is Nguyen Quang, representing the Canton University of Medicine and Pharmacy, Vietnam. Today, I am honored to share with you our latest work, a project that bridges the gap between the green nanotechnological technologies and dermatological care. My presentation is titled Antibacterial Efficacy of Silver Nanoparticle Chitosan Nanocomposite Films against the Acne-Associated bacteria. This research is a fruit of the dedicated collaborations. I would like to acknowledge my supervisors, Associate Professors Lê Hữu Phước and Dr. Trần Thị Như Lê, along with our patient student research. Yeah, their rigorous work in the lab made this finding possible. Let's start with the why. Acne vulgaris is often dismissed as a merged cosmetic nuisance, but the data tells a darker story. It affects 85% of adolescents globally. It is a disease of inflammations, bacterial colonizations, and significant psychological burden, causing anxiety and social withdrawal in the millions of young people. But the real crisis isn't just the disease, it's the treatment. For decades, we have relied on antibiotics. The result? We have first bacteria like Cutibacterium, acnes, and Staphylococcus aureus to evil. We have now facing a resistance crisis. Traditional antibiotics are failing, and they often bring hard side effects. The question we asked were, can we engineer a solution that is effective, safe, and difficult for the bacteria to resist? To do that, we had to know our enemy. We target two key pathogens. Number one, the classic culprit, Cutibacterium acnes, anaerobic. Number two, the opportunist, Staphylococcus aureus, aerobic. As you will see later in my results, as aureus plays a much larger role in acne than previously predicted. Our answers lie in synergy. We designed a nanocomposite film combining the three elements. Number one, silver nanoparticle, the school for non-for patients, broad spectrum killing power. Number two, the Chitosan, the seal, a biocompatible polymer that stabilizes the silver and the remote healing. Number three, alginate, the structure providing the flexibility and moisture. We set out a three clear objectives. Number one, refine the clinical reality of acne patients in our regions. Number two, isolate the wire resistant bacterial strains directly from these patients. Number three, prove that our film can kill these specific clinical isolates. Our study design was unique because it is traditional. We didn't just stay in the lab. We started at the hospital with the patients, moved to the microbiology lab for isolations, then to chemical lab for synthesis, and finally back to the efficacy testing. We enrolled the 65 patients from dilations. We isolated the bacteria using the specific media blood agar for the aerobies and krypton soya agar under an anaerobic conditions for the C. acnes. As you can see on the slide. Okay. Here is where the innovations happens. We utilize the green chemistries instead of toxic chemical reducers. We use a convert fruit extract, citrus, japonica. Why? Because it's eco-friendly, cost-effective, and safe. The the phytochemicals in the fruits naturally reduce the silver nitrate into stable silver nanoparticles. No harsh medical chemicals means a safer product for the human face. We rigorously characterize the material using the UV with SEM and FTIR. Then we set its efficacy using two gold standards. Number one, keep B power for the visual confirmations qualitative. Number two, 96 well micro dilution find the MIC quantitative, the exact concentration needed to stop the bacteria. So let's look at the data. Our 65 patients presented a typical profile, young adults, average 19.8 years, suffering mainly from itching and oily skin. Most had mild to moderate acne. But here is the radical findings. Look at the pathogen distributions where the C. acnes was present. 26.2% Staphylococcus aureus were dominant, found in the 34.4% of the patients. These challenges in the traditional view, acne is our cohort is a bodily microbial infections, driven significantly by staph. This validates our need for the broad spectrum against like the silver. These are the green synthesis work. Absolutely, the UV vis spectrum saw the signatures back at the 438 nanometers. This SEM images reveal the spherical well dispersed nanoparticle with the ideal average size of 37.8 nanometers, small enough to penetrate the material's small walls, but large enough to be stable. You can see the chemical interactions FTIR. Okay. Before we talk about the killing the bacteria, we must ensure we don't kill the skin cell. We perform an NTT cytotoxic assay. The result, our film maintained over 70% cell viability in a human keratinocytes. According to the ISO standards, this is non-cytotoxic. We hit the sweet spot deadly to bacteria, safe for skin. Now, the most important question is work against the patient's derived bacteria. The answer is a resounding yes. Look at the MIC value. low is better. Again, the clinical areas MIC was just 4.23 eurogram microgram per milliliters. Against clinical C acnes, MIC was 4.87 milligram per milliliters. Even against E. coli, my MIC was 5.50 microgram per milliliters. These are exceptionally low concentrations. We achieve potent inhibitions using the trace amounts of silver, minimizing any potential toxicity risks. To summarize the novel insights from this work, we have three novel insights. Number one is re-evaluations. We confirm that areas is a major co-pathogen in acne, requiring the broad spectrum treatment. Number two is the green advantage. We proved that you don't need toxic chemicals to make the high quality nanobacterial materials. Number three, clinical the robustness. Our film works just as well on acquired clinical strengths as it did on the lab strengths. This suggests our chemical mechanisms of the actions physical description of the cell world is immune to traditional antibiotic-resistant mechanisms. Okay, in conclusions, we have successfully synthesized a stable ring-aged Chitosan composite. It is potent against the resistant clinical bacteria, safe for human cells, and sustainable to reduce. This opens the door for the immediate clinical translations. We envision this material being used in the invisible acne patches, bioactive facial marks, or even post-operative wound dressing to prevent the infections. I would like to thank you, my university, and all the patients who participated. Thank you for your attention. Thank you for the M-Nano 2025 to access my presentations. And I welcome any questions you may have on my Gmail below. Thank you and goodbye.